Okay, welcome back to class. We're going to go over research methods in behavioral neuroscience today. And these pictures behind here are just from a bunch of fancy, high-impact neuroscience papers. And so I just picked these because we see, we're not going to go over all of these techniques today, but you can see like we have some calcium imaging here on this little diagram. We have some kind of behavioral assay here. And that's essentially what behavioral neuroscience is it's the combination of looking at the nervous system in a variety of different ways, and trying to tie activity in the nervous system to behavioral states. So this is behavioral neuroscience. I mean, we call it behavioral neuroscience today, but the reason why the class is called biological psychology was because this is essentially what it's evolved into. So initially, you had psychologists who started to become interested in a variety of different biological aspects of behavior. And so as the field of psychologists who were studying brain and behavior relationships grew, we then got our own kind of branch, which was called biological psychology, and eventually just kind of evolved into behavioral neuroscience because the tie to psychology started to seem a little bit more nebulous. You also had a lot of physiologists who came to biological psychology from the other direction. So you had people like Pavlov, who we've talked about and we will talk about a lot in the learning section, who discovered classical conditioning or associative learning, which is one of the hallmarks of psychological studies. But he was a physiologist. He was not a psychologist, right? So you had a lot of physiologists kind of migrating to how physiology in the brain affects behavioral outcomes, because what they cared about was like feeding and consuming fluids and things like this. And then a natural consequence of trying to figure that out is going to lead you to the brain, right? Similarly, a natural consequence of trying to figure out psychological phenomenon, although eating and drinking is kind of psychological phenomenon, I think that what we may typically think of psychological phenomenon is like anxiety, or why people are nervous when they go and talk to strangers, or whatever a variety of psychological assays you care about or phenomenon you care about, it all leads back to the brain. So at its heart, what behavioral neuroscience is the study of brain and behavior relationships, how the brain produces behavior, a variety of types of behavior. And behavioral neuroscience is studied in both humans and animal models. Behavioral neuroscience specifically, though, a lot of times, behavioral neuroscience utilizes both animal and human models. So when I say model in a scientific setting, just in case somebody has not taken like a philosophy of science course or has ever heard of the term scientific model, a model is a tool used in science that represents a real world phenomenon, but is not the real world phenomenon. So if you say a human model, right? Like, why would a human model be a model? Why wouldn't it just be the real phenomenon? Well, it's because if you have, if you want to study drug addiction, or you want to study schizophrenia, or you want to study anxiety disorders, or you want to see what happens to people when they look at a gross image or something, this is a model because it's likely going to be in a context or a setting that is not a natural environment. So if you have somebody who is suffering from substance use disorder, you're likely not going to be able to get an experiment that's going to 100% replicate a real life scenario. And if it does 100% replicate the real life scenario, then it's not going to be an experiment anymore. It's then going to be some kind of observational approach, right? So you could just watch people who suffer from, cocaine use disorder and see what they do, but you're going to be very limited on what you can actually test and things like that. So if you really care about being able to talk about causality, which is what a lot of science does care about, then you need to be able to run an experiment. And the only way that you can run an experiment is by creating kind of a synthetic environment. Okay, so that's why we use animal models, and that's why even if you're using a human, it is still a model of the phenomenon, because as we can see right here, this is a picture of somebody going into an fMRI, which we'll talk about at the end, which is functional magnetic resonance imaging. Even in this scenario, although it's a human, it's a model of whatever they care about, because being put into an fMRI machine and being shown a picture of something disgusting and then seeing what areas of the brain light up is not the same thing if you were walking down the street and you stepped in poop and they're trying to disentangle the circuits that are important for the feeling of disgust, right? So anyway, hopefully that made sense. but models are used to figure out how the universe works, right? And in behavioral neuroscience, we use both human and animal models. You should know what a model is for the exam. I'm likely going to ask a question about that. A model is a scientific tool to replicate a real life phenomenon in a laboratory setting. That's what a model is. So yeah, you'll have to know that. I'll almost certainly ask a question about that. And the reason why you need to know that is because if we're going to talk about research methods in biological psychology or behavioral neuroscience, you need to know what a model is, because that is the predominant method to study phenomenon. Okay, so first we're going to talk about loss of function studies. And while loss of function studies can happen in both animal and human models, when it's happening in a human setting, it's very frequently due to some kind of medical problem. Either something happened that was not, you don't want to have a loss of function study in a human, because what that means is that they've had some horrible damage done to their brain in some way. But so we're going to go over several different types of loss of function studies. The first is going to be observational loss of function studies, and that's going to be primarily what happens when we are able to do these kind of studies in humans. Then we're going to have lesion studies where you actually destroy a part of the brain intentionally due to some kind of experimental manipulation. Obviously that happens in animal models. And then we're going to have pharmacological inactivation, which is using drugs, chemogenetic inactivation, which is using viruses or transgenic approaches to produce some kind of receptor that's sensitive to some kind of drug that you can manipulate the brain with. And then we have optogenetics, which is similar to chemogenetics, except for it uses light as an agonist instead of a ligand, like a drug ligand or a chemical ligand as an agonist. So we're going to get deeper into all of those. And, this is going to be somewhat of a dense lecture in the sense that I don't know how long it's going to be, but it's going to probably cover some ground that you're not super familiar with. All of the vernacular or the vocabulary that I'm going to be using will have been covered in the first 4 lectures that we've done. So I think the first 4 lectures had to do with action potentials, like cell physiology, and chemical events at the synapse, right? So although a lot of this is going to be really applying all of those concepts that we learned in those first 4 lectures, some of what we learned in neuroanatomy is going to be applied here. So I know that it's going to be somewhat dense. I'm going to be using terms that you haven't heard, again, like chemogenetic and optogenetics. But, I'm going to try and be very clear about what you need to know on the exam so you know what exactly to study. So don't worry, it's all going to work out. So the first thing that we need to talk about are lesion studies that are observational, or loss of function observational studies. So Phineas Gage was somebody that I think we talked about in our first lecture, and you have almost certainly taken an introduction to psychology in which case they very likely talked about Phineas Gage. Phineas Gage was a railroad worker who, through some horrible accident, I don't exactly know how it happened, why this thing exploded into his head, but he basically was putting in railroad spikes into a railroad track, and either I don't know if they used some kind of explosive to do this or whatever, but somehow the railroad spike, as you can see through this image, just shot up through his head. It went into his cheek, severed his eye, and then popped out his prefrontal cortex, which is what you can see from this damage right here. And essentially, actually, it went through. Oh wow! So if this image is accurate, basically the story is that he lost a huge chunk of his cortex. But likely, if he was impaled in this section of the brain as well, then he lost a lot more than just prefrontal cortex. Let's say that. But in any case, the point is that he lost a large chunk of his brain, and his behavior changed almost immediately as soon as he recovered, that is. So he before that was very conscientious. He was a very respectable, very nice person. And then after he lost this big chunk of cortex, he became a jerk. He would cuss a lot, and he was very hard to be around. He was irritable, etc. So he had like this really stark behavioral change. And this was kind of, these kinds of studies, and this study in particular, this wasn't a study, but he was then studied after it happened, which is how these things normally occur, was kind of the birth in some ways to behavioral neuroscience, because it is quintessential technique in the field of behavioral neuroscience, which is that if you're interested in some behavioral output like personality or mood or a variety of behavioral outbursts or some anything that you can think of as a behavioral output, which most things are when it comes to conscious creatures like us, you know on a fundamental level that it must be due to some area of the brain. So the easiest way to tie a brain area to a behavior is by destroying that area of the brain and then seeing if the behavior persists or changes. If you were interested in disgust and you had the feeling, or you had some evidence rather, that the amygdala was important for disgust, and you remove the amygdala, and now the person no longer feels disgust. It's like, okay, well, we can say with some level of confidence that the amygdala mediates the disgust response, or whatever we want to call that. So Phineas Gage was one of the first observational loss of function studies. And loss of function, I'll just go ahead and define it, is where essentially you are trying to produce a deficit in the behavior by destroying or inhibiting or shutting off some area of the brain. In this case, it was permanent. Another example of a loss of function study would be with HM, who we will go over in greater detail in the learning module. But HM had I forget what H&M stand for. I think it was Henry Malaise or something like that. But basically, the point is that HM had a part, or actually almost his entire hippocampus removed. So he had a very severe seizure disorder, which was making his life kind of impossible to live. So the surgeon thought that one potential fix for that would be to remove temporal lobe, and essentially he removed so much of it that he basically removed all of this guy's hippocampus. And so he had severe anterograde amnesia, which is that after the event occurred, he could no longer make new memories. So that was one of the first studies, to my knowledge, that showed how important the hippocampus was to learning and memory. So, this is a very typical type of situation in behavioral neuroscience. In this case, they are case studies. They're not actual experimental studies or even really observational studies. These are case studies where they see an individual with a particular phenomenon occur, and then they look at that phenomenon, and then it kind of drives hypotheses, and then we can test it experimentally. And indeed, people that have cortical damage do have affective changes and behavioral changes and difficulty regulating control and things like that we saw with Phineas Gage. So moving on, these are still under the umbrella of loss of function. And now we're talking about lesion studies. So let's see if I have that. Yeah, so we have lesion studies next. And there's a variety of lesion studies. The most common are electrolytic and excitotoxic. So electrolytic are lesions that are produced through electricity, like they basically take some kind of I don't even know what you call it, a lead of some sort, and they will fry an area of the brain with electricity. So this is a picture from a study, I think it was in the 80s, where they were interested in the dorsal hippocampus and trying to figure out if the dorsal hippocampus was necessary for contextual learning. And so here you can see, this is, I don't know if this is a rat or a mouse brain. It's kind of hard to tell because of how old their picture technology was. I don't know if, like, what kind of microscope or what kind of microscopy this is, but it's a little bit hard to tell, but this is definitely either a rat or a mouse. And essentially here, what I'm highlighting, you can see that there's just a giant hole where the majority of what's called the dorsal hippocampus should be. Over here as well, they didn't get as much of the dorsal hippocampus removed, but nonetheless, they still have a big hole here. And so essentially, just like with Phineas Gage, the goal here is to see if by destroying with a physical injury via electricity, will the rodent have trouble learning in a contextual way? Like, will they be able to solve a maze? Will they be able to remember which side of the maze the cheese was on, etc.? So that's electrolytic lesions. Another kind of lesion study is excitotoxic lesions, where you inject an excitatory neurotransmitter that causes cells to be hyperactive and ultimately causes cell death. So this is referring to injecting excessive amounts of glutamate, or causing glutamatergic neurons to be hyperactive, and overstimulating glutamate results in cell death. So, and it's the glutamate itself which is cytotoxic. So this is just another way of producing a lesion, just like the electrolytic lesion technique is, except for this is just using a chemical or a neurotransmitter or something else to cause the damage, rather than actually just using a physical stimulus like an electrode or a knife or whatever. So this is I'm just reiterating here that this is also called loss of function, and this is primarily done in animal models, as you would expect. I mean, we can't morally experiment on humans in this way, of course. So if it is done in humans, though, it's due to medical accidents or conditions, like we already said. You can have loss of studies or lesion, like this is essentially a lesion study here, although it is not very granular. It's primarily done in animal models, and if it's done in humans, it's due to accidents, and it's going to be in an observational context. It's not going to be in an experimental context. It'll be a case study or something like that. So what you need to know from this is you definitely need to know that electrolytic lesion studies are using, like a physical electrode to produce electricity to then destroy a part of the tissue. You need to know that excitotoxic lesions are using chemicals or other excitatory neurotransmitters to produce the lesion. And what's actually happening is that you are just removing a piece of the brain, or you're destroying a piece of the brain. The next technique, and also like the way that I'm kind of describing these studies is kind of in chronological order. All of these techniques can still be used in contemporary neuroscience, but like lesion studies are not used nearly as much as the next several types of techniques that I'm about to describe. And that's just because a fallback of lesion studies is that they're hard to control. So you don't have the kind of granularity or specificity that you will get in the next one. And in fact, that will be a test question, I'm almost certain, is that a drawback of lesion studies, and basically any kind of loss of function study from, a long time ago, is that they're not very specific to the neuronal population that you care about. So just make sure to know not only that electrolytic lesions are produced with an electrode through electricity, and that excitotoxic lesions are produced through chemicals or excitatory neurotransmitters, but a drawback of using those techniques is that you don't have very good resolution, which means that, while we do get rid of the entire dorsal hippocampus here, we also are messing up some of the brain below the dorsal hippocampus, which reduces our ability to, confidently say that it's the dorsal hippocampus and not some other region. Also, you can see here that it's not very consistent. Like the electrolytic lesion that was produced on this hemisphere of the brain was a very different shape, and it didn't get as much of the hippocampus. It got some other area that's not the hippocampus, right? While this one was primarily the dorsal hippocampus. So if this rat had a deficit in some behavioral output, then how do you know it's because of the lesion to the left hemisphere and not the lesion that was to the right hemisphere? And I'm saying left and right because this is the left side of the rat's brain. So that's obviously not going to be an exam, but like when the brain is facing you this way, the rat's left side is on your right. So anyway, then we have pharmacological inactivation. So pharmacological inactivation is very similar to a lesion study with electrolytic or excitotoxic lesions, except for they are very often temporary. So if you were to do some kind of study where you just destroy or ablate a bunch of dopaminergic neurons, that would more so fall under the categorization of some kind of cytotoxic lesion rather than a pharmacological inactivation. Pharmacological inactivation is the use of drugs to temporarily inactivate a brain region. It can be more useful than lesion studies because instead of irreversibly destroying the brain area, it will come back online once the drug wears off, allowing you to do multiple tests in the same animal. So that's the other thing, is that with this lesion study here, once I destroy the dorsal hippocampus, it's not coming back, right? So I can't, test the animal's ability to solve the maze, then destroy the hippocampus, test the animal's ability to solve the maze, and then, bring the hippocampus back to see if the animal can solve the maze okay, right? Like once I destroy the hippocampus, it's gone. So using a pharmacological inactivation method instead is, the benefit, other than having greater specificity and not just like wrecking a whole big portion of brain, is that you get to have the drug wear off. So, if we have some brain region here, so the big circle is a brain region, all the little circles are neurons within the brain region, and we can use a cocktail of drugs. In this case, I'm giving you mucimol and baclofen. These are GABA A and GABA B agonists, respectively, and they bind to the GABA A receptor, the GABA B receptor, and essentially what that does is it inhibits activity in that entire brain region. So if you remember, GABA is the main inhibitory neurotransmitter in the brain. So if you activate a bunch of GABA receptors in a particular area of the brain, there's going to be a lot of inhibition, which means that area of the brain is going to stop working. Now, as soon as the drug wears off, of course, now you can retest the animal in the same behavioral assay that you wanted to do, and now they won't have that pharmacological deficit that was there previously. So what do you need to know about this? You need to know the benefit of pharmacological activation is that you have higher specificity, which I don't have written down, but you should know. You also have the fact that since it's pharmacology, it's reversible, right? So the other lesion studies are irreversible, but pharmacological inactivations are reversible. I was making sure that I had that written correctly here. Okay, so this part might get a little bit more confusing, but I promise if you understand what a receptor is and what an agonist is and what a ligand is, ligands are just agonists, just another word for that, then I think that we'll get through this okay. So chemogenetic inactivation is referring to very more often than not something called dreads. Chemogenetic does not need to mean dreads, but it very frequently does in behavioral neuroscience. So in the context of this class, chemogenetic and dread is basically synonymous. DREADS is an acronym for designer receptors exclusively activated by designer drugs. So essentially what this means is that they take a synthetic receptor and they take a drug that is specific to that receptor and activate that receptor. And so the big benefit of a DREAD, inactivation or loss of function study is that you have very high specificity. Okay, so I want to say that again because this is likely like the main question I'll ask about this. Like the benefit of DREADS is that there is very high specificity in the neuronal population that you want to manipulate. So it's typically based off of a G protein coupled receptor. So remember that G protein coupled receptors are also called metabotropic receptors. And they're called G protein coupled because it is a 7 membrane protein. It spans the phospholipid bilayer 7 times. And on the intracellular space, that G protein coupled receptor is coupled to a G protein. What is a G protein? It's some chemical that's residing on the intracellular space. But the important part is that when you activate that protein, so when you have a ligand that binds to it, the G protein on the inside, it becomes decoupled and it goes off and has some downstream effect, right? It could either hyperpolarize or depolarize the cell. Hyperpolarizing meaning getting it away from an action potential, making the cell more negatively charged, whereas depolarizing means producing an action potential or making the cell more positive. So this can be produced through transgenic animals or through viral transfection. And it's important to note that transgenic means using genetic manipulation to add or remove genes that will ultimately produce some kind of outcome. So transgenic is referring to manipulating the genes, either putting in a gene or taking out a gene to then produce something that you care about. In this case, what we're talking about is manipulating the genes in a way to then produce a DREAD, which is again a synthetic G protein coupled receptor, right? So it's a protein that is made in a lab, and you can manipulate the animal's genome such that they express this DREAD at a particular brain region that you care about. So you can make it express in a particular subset of neurons. So you could say, only in dopamine neurons I want to express this DREAD. You could also say, only in serotonin neurons I want to express this DREAD, or whatever. You can also do it through viral transfection, which is another way to manipulate genetic expression. It's just that instead of breeding it into the animals, which is what transgenic manipulations are referring to, you're injecting it into the animal after the animal's already alive and grown, typically. So those are the two ways that you can induce a DREAD, is that you can do it through transgenic manipulation by breeding the animals in a very particular way, or you can take what's called a wild type animal, meaning the animals that have no transgenic manipulation, and you can inject a virus that will then replicate itself and cause genetic changes, or basically transfect a subpopulation of cells and produce the DREAD. Okay, so what you're looking at here is a diagram of a DREAD that's being used. I'm just trying to, because this is not as complicated as it might look. Um, so I would like to ask a question about this on the exam, but probably it's getting a little bit too granular. So I won't like have a question this in depth. But it's just good to know that what you're looking at right here is a viral tool, and this, like all of this acronym right here, is explaining different parts of this virus that these researchers were injecting. So the reason why I'm showing you this is because this is a dread, right? So it's kind of hard, like if you don't have the context for what a dread is, it's like it's a synthetic receptor, but in practice when it's used, oftentimes you'll see a dread in a paper written in this way. And so what this all means are essentially, it's telling you how they're getting the dread into the animal's brain. So this is being put into this is a viral transfection where they have an adeno-associated virus. That's what AAV means, adeno-associated virus. And then they have this promoter here. So this is an MCH promoter. The promoter of the viral sequence is telling you which neurons are going to express the dread. So in this case, it's melanin concentrating hormone. And then they tell you what kind of dread they have. So like I said, a dread, which is a designer receptor exclusively activated by designer drugs, is a synthetic receptor, meaning that the researchers took a real receptor, in this case a human muscarinic receptor. If you remember, muscarinic receptors, actually that's not fair because I don't think I told you guys this, but a muscarinic receptor is a G protein coupled receptor. It's sensitive to acetylcholine. And they took this human muscarinic receptor and they manipulated it so that it would only be sensitive to a particular type of drug. So instead of the receptor being sensitive to acetylcholine, which your body naturally produces, it's now sensitive to a synthetic drug that you can inject, exogenously. So, and then this is GQ. This means that it's the type of G protein that it's coupled to. You definitely don't need to know this, but just in case anybody happens to know this and is going to get triggered, this is an excitatory G protein, but and I have it under chemogenetic inactivation, but don't worry about that. It was just a really easy, good diagram to kind of show and demonstrate my point. So the next part is that we have this M-cherry thing right here, and this is just a fluorescent protein, so you can actually see where the virus expressed. So this is actually a picture from a rat brain that I injected dreads into for an experiment that I did fairly recently, and you can see what I'm highlighting down here, like all these this little red blotch. This is actually what's called M-cherry, and this is how we see where the dread expressed, okay? So when we put it under a microscope, we can then shoot a particular wavelength of light that reacts with this red fluorescing protein. And then that's how we can say, okay, these neurons got the dread. And again, this is going to be a test question that I've already alluded to multiple times, which is that as our technology has increased, the main benefit of that is the specificity and the granularity of the neurons that we can manipulate. So back when we just had, destroying big areas of the brain, that's not very granular. Here, however, we can say, I only want to inject or infect a very specific subpopulation of neurons. In this case, it was melanin concentrating hormone expressing neurons in the lateral hypothalamus, but this is a huge leap in our ability to test what the brain, and specifically what sub areas of the brain control. So here is a viral vector that is going to express a dread. Oh, so I already kind of wrote this here. Not testing material, but the virus shown in the graphic is technically an excitatory dread, not an inhibitory dread. Yeah, so don't worry about that. That's just in case people are familiar with it and they rightly point out that that's an excitatory dread. But in any case, we inject the dread into the lateral hypothalamus in this case. And what's happening here is that we have a dread expressing neuron. And that dread expressing neuron is going to be infected with the virus that's then going to express this either inhibitory or excitatory synthetic receptor that is sensitive to a ligand of a specific type of exogenous chemical. And then it's going to inhibit this downstream postsynaptic cell when we give it the drug that is selective for that dread. So this is a dread agonist called clozapine N oxide, and it is specific to the dread receptor. So here you can actually see the dread receptor. Like I said, it's a 7 transmembrane protein. It's coupled to a G protein. And in this case, the diagram that I'm trying to show you here is that we have a neuron that's, releasing neurotransmitter. I then inject clozapine N oxide, which is the selective dread agonist, and it inhibits that neurotransmitter release. Okay, so that's what this is trying to show you. So what do you need to know for the exam? You need to know what dread stands for. It's a designer receptor exclusively activated by designer drugs. You need to know that dread viruses and dread transgenic manipulations give us incredible specificity. You need to know that dreads are synthetic receptors, typically G protein coupled receptors. And you need to know that they are activated by a synthetic ligand or a exogenous chemical rather than endogenous neurotransmitter. Okay, so I know that's a lot of jargon. However, we have gone over all of those terms in the previous lectures. So if anyone's having trouble with this section in particular, please email me right away so we can set up a office hours or something like that so I can hopefully break it down a little bit more clearly for you. So moving on to optogenetic inactivation. Here we were talking about chemogenetic inactivation, and that's because we're using a chemical in combination with genetic manipulation to get the effect that we care about. With optogenetic inactivation, it's very similar, except for instead of a chemical, we're using light to manipulate a particular protein. So optogenetics is very similar to chemogenetics. You use a virus or you use some kind of transgenic manipulation to express a protein called an opsin, which is an ion channel that is sensitive to light. And in this case, we're referring to a halorhodopsin, which is inhibitory. So when you have this protein that is an ion channel, it's a light gated ionophore, right? So we've gone over all those terms as well. If we had a voltage gated ionophore, like with sodium channels for an action potential, here we have a light gated ion channel, which in this case is sensitive to chloride. So when they stimulate this protein with light, in this case it's a yellow light, it opens up, it causes a conformational change in this ionophore, which then allows the influx of chloride. When you have the influx of chloride, what happens? The cell becomes more negative, right? So what you can do with this technique is you can actually inject this halorhodopsin into some area of the brain, like the lateral hypothalamus, and you can shine a light, a yellow light, specifically on those receptors or on those neurons, and you can shut off that area of the brain. Now, like I said, the timeline of these techniques that I've been giving you get more and more specific and more, it gives the researcher a lot more control, right? So we start off with these electrolytic lesions, which you have very little control over. Then we have the pharmacological inactivation, where you can inject a drug directly into some area of the brain and it can wear off. And then we have, a synthetic receptor. But the problem with this is that it's still activated by a drug, right? So it still takes time to have its effect. The beauty with optogenetics is that it's with a light, so it happens immediately. You have essentially, I actually showed you a video in the first lecture of a rat, or a mouse, having an optogenetic manipulation where it was eating food. So if you want to see that, go back to that lecture, and it was like the last slide, one of the last slides, where you turn on the light, it shines directly onto a group of neurons and immediately has an effect. And then you can just turn the light right off. So this gives you incredible specificity and control over the manipulation. So what you'll need to know for the exam is that optogenetics, or a halorhodopsin or whatever, an optogenetic approach is using a light-gated protein, a light-gated ionophore, to manipulate cellular activity. So the difference between DREADS and optogenetics is that DREADS use another difference is that they use a G protein coupled receptor that's sensitive to a synthetic drug, whereas optogenetics use an ion channel that's sensitive to light. Again, I know that this can be a little bit confusing, so if you have trouble, go ahead and email me and let's set up some office hours to talk about it. So all of those that we just went over was loss of function studies. So all of these were loss of function. Observational with Phineas Gage, lesion studies where you're just ripping out a piece of the brain, either chemically or electrically. Pharmacological inactivation, where you're using drugs to temporarily inhibit an area of the brain. Chemogenetic inactivation, where you're using DREADS, synthetic receptors, to temporarily inactivate an area of the brain. And then optogenetic inactivation, where you're using a light-gated ion channel to temporarily inactivate some area of the brain. Now, gain of function, as you might guess, is literally the exact opposite of the loss of function. You excite or you cause some area of the brain to become more activated during a period of time, and you see if that causes the behavior that you care about. So you have both chemogenetic activation, which is using DREADS and an excitatory dread to cause neurotransmitter release. You also have optogenetic gain of function studies, where in this case you use what's called channel rhodopsin to stimulate these ionophores, and in this case it will allow the influx of sodium instead of the influx of chloride. And you just do all the same studies that you would do with the loss of function, except for you do it with the gain of function. So you stimulate the dorsal hippocampus and you see, do the rats learn the maze faster? Or when I stimulate this area of the brain, does it cause the rat to pee? If I stimulate this area of the brain, does it cause the rat to spontaneously consume food, etc.? Whatever behavior it is that you care about, you inject the DREAD or the optogenetic, you stimulate it, and then you see what happens to the behavioral approach. That's the gist. Okay, so I'm not going to go super in depth about electrophysiology. We have a couple of slides here, it looks like, on ePhys, but electrophysiology is a complicated area of neuroscience because it has to do, again, with action potentials. So we'll try and keep this shallow, surface level. But electrophysiology, broadly speaking, is the study of the electrical activity of the body. And in neuroscience, obviously, the focus will be on the neurons. So here, again, we have our diagram of the action potential. I'm not going to go over this again. But what electrophysiology is studying is basically how do action potentials occur? What kind of channels are being used to cause the action potential? Is it sodium? Is it potassium? Is it chloride? And it just gets very complicated very quickly. What I talked about and what this diagram is showing only has to do with sodium channels and potassium channels, but what it doesn't show us is a variety and how many sub categorizations there are. So it's only saying potassium channels here, but there are dozens of potassium channels. There are dozens of sodium channels, and they all are opening and closing at different points to propagate the action potential. So electrophysiology tries to link the electrical activity of the neuron with the behavioral output that we care about. There's two types of electrophysiology. This part you will have to know just because it's very helpful in biology or biomedical sciences in general to know the difference between these two. But ex vivo means, so I'll just read it so I don't try to read it and extrapolate at the same time. But ex vivo and in vivo are common terms used in research. Vivo is a Latin term in this context meaning in the living or life, and ex vivo refers to tissue that was taken from an organism that is now dead. In vivo refers to studying the phenomenon in a living organism. So you also have in vitro, so this is not to be confused with in vitro, which is referring to cell cultures, but whenever we're talking about vivo, whether it's ex vivo or in vivo, we're talking about research that was done in an animal that is either dead or alive, right? So it's if you're doing ex vivo, that means that you removed brain tissue and now you're studying the brain tissue now that it's out of the rat's head. Or you could do in vivo, which means that you're studying the tissue while the organism is still alive. So ex vivo electrophysiology is, as I already kind of described, where you remove the rat's brain or the mouse's brain and you keep the neurons functional so then you can test that. So let's see what my bullet points are. Some behavioral measurement is done. Yeah, so maybe you do some kind of like contextual fear conditioning where you scare the animal, or maybe you do some kind of operant self-administration where the animal is giving itself cocaine or whatever. The rodent is euthanized and their brain is removed, right? So, some behavioral measurement is done. You kill the rat and you take the brain out. The neurons can be kept alive in artificial cerebrospinal fluid. So, as we talked about in the last lecture for neuroanatomy, the brain is basically surrounded and filled in the ventricular space with cerebrospinal fluid. So you can actually have artificial cerebrospinal fluid, which has all of the same things that real CSF has, but it's outside the brain, and you put the brain in it to keep the neurons functioning. Researchers take measurements of electrical properties of the neuron that may have been manipulated by the behavior, the drug, or the manipulation of interest, and use a glass pipette to measure the electrical activity of the cell. Yeah, so this is actually, it's a little bit complicated, but it's incredibly fascinating. You take a glass tube, basically. That's what a pipette is, a glass pipette rather in this context, and you stretch it out so it's so thin that it actually is small enough to puncture the soma of a neuron. So this is incredibly small, and it's hilarious because I did a rotation in graduate school in an electrophysiology lab, and the pipette tip is microscopic. Okay, so literally you can't with the naked eye see the tip of the pipette that you're going to be puncturing the soma of the neuron with. And so a lot of times in grad school what would happen is that I would accidentally break the pipette tip but not know until I saw it under the microscope. So it's like it takes forever to get the pipette. You put it in, you finally find it under the microscope, and then you see that it's like jagged, so you have to start over. But basically what this does is it creates a contiguous surface with the soma of the neuron so that you can then measure the activity of the cell. And that's how all of these studies that are, talking, like how do we even know about the influx of sodium and a increase in charge of the neuron? It's because you can directly measure it with ex vivo electrophysiology. There's also in vivo electrophysiology, which is where you have electrodes that are implanted into the brain of an alive rodent. And here are some of the pictures of the different types of electrodes that you can have. You look at some kind of behavior or drug manipulation, and then the electrical activity of populations of neurons are recorded. So the difference here is that instead of having, being able to see what one neuron in particular is doing, here you can look at entire populations of neurons. So the differences here are that ex vivo allows greater specificity on a cellular level, but the animal is dead, which limits what kind of manipulations you can do, of course. And the brain region is now isolated from the rest of the brain, which may change its function, right? So it's like if you are just recording activity from an amygdala neuron, it's hard to say what that amygdala neuron would be doing if it still had the activity or the innervation of the rest of the brain. In vivo means the animal is alive, giving you greater behavioral control, but you have less resolution on the cellular level. So for the exam, what you need to know about electrophysiology is that one, it's the study of the electrical activity of the neurons. And two, you need to know the difference between ex vivo and in vivo electrophysiology. Ex vivo is electrophysiology done on a singular cell that is outside of the rodent, whereas in vivo electrophysiology is done on a multiple cells in an alive rodent. So you should also use this slide, these 3 bullet points, these 2 for ex vivo and this one for in vivo, as your studying parameter for this section of the exam. So next we're going to go over the behavioral studies, which thankfully is not typically as hard to digest as all the other stuff that we just went over. We only need to know about 3, but just know that behavioral studies, while it tends to be a little bit easier on the surface to understand, they actually can get very complicated. We're not going to go over any complicated ones, but let's just say that there are some behavioral tasks that can take 6 months to a year to teach the animal to do properly. So it can be a nightmare because your experiments will take forever. I mean, if it takes 6 months to teach the animal how to do the thing, then you're going to be sitting there for a very long time trying to generate data. So the 3 that you need to know are condition place preference, also called CPP, self-administration, and a 2-ball choice test. So the first one, condition place preference, is used in drug studies, but it can also be used when testing if certain stimuli have aversive or rewarding aspects. The main dependent variable in a condition place preference experiment is the amount of time spent on one side of the chamber versus the other. And by main dependent variable, what I'm talking about here is the thing that you are interested in observing. So if you care about whether or not a drug is aversive or rewarding, you will infer that based off of how much time the animal spends on either side of this chamber. So with condition place preference, this is exactly what it looks like. You literally have this 2 little, rat or mouse houses, and you have distinct characteristics to each chamber. The animal is then placed in one chamber and given a drug or some kind of stimulus that you care about measuring its rewarding or aversive aspects. And then after several days of pairing one side of the chamber with your stimulus, you then put the rat or the mouse in the center chamber and allow them to run freely in between each space. If the drug you gave them was aversive, then the mouse or the rat will avoid the chamber where it got the drug. If it's rewarding, like cocaine or some kind of drug of abuse, then the rodent will spend more time in the chamber where it got the drug. And this was one of the first ways that we discovered that drugs of abuse are rewarding to all animals, including rodents, because they were giving cocaine to mice and shining a light directly on the chamber where the mouse was getting cocaine, and the mouse would spend more time in the light chamber, which rodents do not like to do. They like to go towards the dark. They are skittish and afraid of you, so they try to get away. They go towards the corners and they get away into the darkness. But giving a mouse cocaine injections in a light chamber, the mouse was cool with it. Spends a lot of time in there. So this is condition place preference. What you need to know about this is it's a way to commonly study if drugs are aversive or rewarding. It is a Pavlovian or an associative learning procedure, but you'll learn what that actually means in our learning section of the course. Let's see, anything else for this? It can be both aversive or rewarding effects, but basically you're measuring how much time the rodent spends on either the drug paired or the non-drug paired chamber. And I'm saying drug paired because I studied drugs of abuse in grad school, so I'm kind of biased to saying that, but it could also be with food, you know what I mean? Or it could also be with an aversive stimulus. So that's condition-placed preference. We also have self-administration, and self-administration is done commonly in all sorts of learning studies. And typically with learning studies that have a rewarding stimulus. So in grad school, all of my data was generated from self-administration of cocaine and oxycodone. So you would train the rat to get an intravenous infusion of either cocaine or oxycodone. This is a picture of a rat in a operant box, very much like the one that I used in grad school, except for it isn't hooked up to any kind of intravenous injection system, which means that this must be a some kind of a food study. So basically what will happen is the rat will go up to this little lever right here, press the lever, and then this light will turn on and a food pellet will be delivered. So the rat will learn that when I press the lever and see the light, then I get a food pellet. And they love to do this. They will hoard the food pellets, even if they don't eat them all. I don't know why, but they'll just get a whole cache of food pellets here and hang out with the food pellets. And the main dependent variable in self-administration studies is the number of times the rat presses the lever. So I said active lever here because generally there's two levers, one of them that does a nothing, and then one of them that actually gives them the reward. And the reason why you have two levers is because the inactive lever is just to see, is a control to see if the rat will press, just likes pressing the lever, right? So if they press the inactive lever and the active lever the same amount of times, like this rat is just kind of an idiot, likes pressing levers, you know? So, okay, so that's self-administration. You should know that it is a Skinnerian or a operant learning procedure. It involves pressing a lever and getting some kind of rewarding stimulus. It could be drugs, it could be food, but that is what self-administration is. And just like the name, it's just how the name sounds. It's self-administration. There are also human studies that use self-administration, but typically, if you go up to a behavioral neuroscientist and you say cocaine self-administration, they're probably going to think of this rather than a human study. In human studies, though, they do happen. You can pay participants to stop using drugs, and so they'll, give the people like a crack pipe, and they'll say, you can take a hit off of this crack pipe, or I'll give you $5. No joke, that is a type of study that happens, and that is a self-administration study because the person is administering the drug to themselves. But in the context of this course, I'm going to be referring to rodent self-administration. So we also have two bottle choice tests, and this is just like what this picture suggests here. You take the rodent, you put it into a chamber, and there's two bottles that the rodent can drink from. In this case, it's saccharin and H2O, water. And the saccharin is a sweet flavored, they're just putting saccharin in the water. So in this scenario, they would be testing if their manipulation affected how much the animal drank water compared to saccharin. So the main dependent variable in these studies is the amount of solution that the rodent consumes from one bottle compared to the other. So this is very similar to condition-placed preference. Instead, though, of there being a chamber that you're testing, in this scenario, you're testing the ingestive behavior of water or saccharin. So, for this, you just need to know exactly what is here on this slide here, that it is, testing how much of one solution the animal's consuming over another solution. And that could be something as mundane as like, well, is this solution reinforcing? Does the animal like it? Or it could be something more complicated where maybe they destroyed some kind of hedonic or some kind of reward center in the brain, and they want to see if this mouse in this study still likes saccharin even though it can't feel reward. And so you could, oftentimes what will happen in behavioral neuroscience is that you combine these behavioral studies with these more complicated, chemogenetic or optogenetic studies. So you might, put optogenetics into this mouse's ventral tegmental area, stimulate it, and see if that affects how much saccharin it drinks over water, for instance. Okay, so almost done. Just 2 more to go. fMRIs are functional magnetic resonance imaging techniques. They are used primarily in human research. They are sometimes used with animals, but typically it's with humans that you see this kind of thing done. It shows areas of the brain that are active, and it does so by using a high-powered magnet to look at blood flow. So it can track where essentially there are high areas of blood flow and look at that, and that is used as a proxy as neuronal activity. So as I kind of already talked about in the very beginning, maybe you want to study disgust in humans. And so you put somebody into an fMRI machine, and then you show them a picture of poop or vomit or something, and then that causes the amygdala or some cortical region to have high areas of blood flow. And so then you can kind of infer that, well, this area of the brain is highly active when I'm showing them this disgusting image, therefore this is a disgust area of the brain. Obviously that will come down to interpretation, but whether it's disgust or maybe they're annoyed or maybe they just maybe they even like it, who knows. But the typical dependent variable with this is blood flow as a proxy for neural activity. So obviously, there are pros and cons, as I already said, to rodent research and to human research. The more your model deviates from the thing that you care about, the less applicable your findings will be to the real world, right? So, one critique of animal research is like, well, I mean, if you cared about drug use and substance use disorder, like, can you really study that in a rat? Like, are rats actually drug addicts or, individuals that suffer from substance use disorder? I would say no. You know what I mean? So you're losing some applicability there because you're not actually studying drug addiction, but you are studying a part of the physiology that must contribute to drug addiction, right? Similarly, in a scenario like this, you're using humans, so that's a plus. It's like at least it's the same species that you care about. But when you're putting a person into an fMRI machine and you're showing them a crack pipe, is that really the same thing as when somebody's out there seeking crack? It's not really the same thing. So you are losing some translational value no matter what kind of scientific model you're using. So I just want to comment on that. Okay, so for an EEG, which is an electroencephalogram, this is where, just like this picture shows, they put electrodes all over your head, and they can look at the gross neuronal activity that's happening. So you can see the overall electrical activity in the brain. It can be used to diagnose pathologies like epilepsy and sleep disorders, but it does not have the resolution that fMRIs do. So you can't really see exactly, it's in the amygdala here that we're getting this activity or whatever. So you should know the difference between the EEG and the fMRI. One being that, one of these is using blood flow. The other one is just looking at gross electrical activity. And that'll probably be like the main question that I ask. I will also, so we're at the end of the lecture. I'm just now kind of thinking about questions that I'm most certainly going to be talking about and asking on the exam. I will also want you to kind of be able to answer why there are limitations to animal studies, why there are limitations to human studies, and why just in general scientific models have potential limitations but also benefits. You know what I mean? So like I said, a limitation to a human study is going to be that it's very often the case that you can't experimentally manipulate the real life scenario. For instance, you can't go out and shut off somebody's dopamine release and then let them smoke crack and see if that actually stops them from smoking crack, right? Like you can only let them smoke crack and then look at activity of the dopaminergic centers of the brain and see if there's a correlation between that. You can't actually do an experimental manipulation where you shut off the dopaminergic production and see if that stops them from doing the drug. Whereas you can do that with a rat. Okay, so you can actually shut off the rat's dopamine center and see if they still want to do cocaine. The downside to that is that you're using a rat, right? Like a rat's not a human. So, and we don't care about rat substance use disorder. Unfortunately, maybe we should. But in any case, so this was methods in behavioral neuroscience. I know that one was probably pretty dense. If people, need some more time with this and want to go over some of the, dreads or optogenetics or whatever, please email me so we can talk about it. It's not like I'm going to have 50 questions on these things. Just know, in general, what the concept was, and I think that you'll be fine. I also will probably be spelling a lot of this out in the questions themselves. So I'm just saying that so people aren't like preemptively freaked out and emailing me saying that there's no way they can learn this. I'm pretty sure that most of you are going to do pretty good on this. So in any case, I will see you in the next lecture, which is, I believe, psychopharmacology. So until next time.